My name is Ashley Collins, and today I'll be discussing the differences in lean versus obese gut microbiomes. In this presentation, I will introduce you to what obesity is and what the gut microbiome is. I will be talking about lean versus obese gut microbiomes and the studies used to show the differences between them. And I'll be talking about what firmicutes are and their role in obesity. Obesity is defined as having a body mass index of 30 or higher. Greater than 1/3 of adults are obese in the United States, making it a growing health problem. Obesity is associated with multiple health conditions and increases the risk of them. Some conditions include heart disease, diabetes, and stroke. Type 2 diabetes is actually the condition that is most heavily influenced by obesity, and 90% of type 2 diabetes patients are overweight or obese. Many factors can contribute to becoming obese. Food choice, genetics, exercise patterns, and caloric intake are some, but there are thought to be many more. The microbiome actually has become a growing topic of interest within the last decade. The microbiome is defined as the combined genetic materials of the microorganisms living on and inside the human body. Every microbiome is very unique to the individual, depending on genetics, where that person lives, the type of diet that person's on. The microbiome plays a part in many of the body's natural processes. It can promote functions of the immune system, play a role in metabolism and digestion, which is what I'm going to be talking about today, affect insulin sensitivity, assist in detoxification processes, and can even play a role in the health of a newborn baby. So, like I was saying before, the microbiome is thought to be correlated with obesity. So, before going into the studies that I'm going to talk about, I just wanted to talk about the fact that studies have been done on both mice and humans. The ones I'm going to be talking about are on mice, but it has been shown that they are a good subject. They have greater than 90% of their bacterial phylogenetic types in the gut are of two bacterial divisions, the firmicutes and the bacteroidetes, and that is also the same as in humans. Studies in humans and their gut microbiomes have actually shown similar results, making mice an ideal species to test this on. In 2008, Turnbaugh, Blackhead, Fulton, and Gordon conducted an experiment to analyze diet-induced obesity in mice. They started off with 10 germ-free mice, essentially meaning these mice had been born and raised in a sterile environment that was free of microorganisms. At 12 weeks, each mouse was given the gut microbiota from a conventionally raised mouse. So basically their microbiome had been transplanted by a mouse that had been raised conventionally, meaning there had been microorganisms within its environment. This was to ensure that each of these mice had started off with the same microbiome. At 16 weeks, half of the mice stayed on the low-fat diet that they had started off with. So as they were born, they were given a diet that was low in fat, and this is going to be called the chow diet, or CHO. And the other half of these mice were put on the Western diet, which is high fat and high sugar. The fats in this diet are saturated and unsaturated fats, so a combination of both. And the sugars are carbohydrates used in common food additives. And this diet actually, 41% of the calories are from fat. And so these mice were on these two diets for 8 weeks, and afterwards full-length 16S rRNA sequences were taken of their gut microbiomes. So the mice that were on the Western diet gained significantly more weight than the mice that were on the chow diet. They had higher ratios of firmicutes, so the mice that were on the Western diet showed a greater ratio of firmicutes than the mice that were on the chow diet. And the mice that were on the Western diet had a lower bacterial diversity. So So their microbiome showed that firmicutes were overpowering the other branches of bacteria that compose of the gut microbiota. So here's a better explanation of the results I just shared with you. Figure 1 is showing the percent of total 16S RNA sequences. So it's basically showing the firmicute and bacteroidete ratios. In the chow diet, as you can see here, there is more of an abundance of firmicutes than bacteroidetes. However, bacteroidetes are present here, and the firmicutes aren't taking up the whole entire microbiome. Versus the Western diet, you can see that firmicutes is almost 100% of this microbiome sequence, and the bacteroidetes are basically not present. There is such a low number of them in comparison to the chow diet. Figure 2 is showing the percentage of mollicutes. Now, mollicutes are a specific branch of firmicutes. So this branch of firmicutes has the highest abundance. And as you can see, the Western diet mice have a high percentage of mollicutes compared to the chow diet. And figure 3 is showing the percentage of increase in body fat. And again, Western wins with a higher percentage of body fat increase than the mice that were on the chow diet. So in this diagram here, it's showing you in experiment A, what I had just explained in the previous slides. The germ-free recipient was given the chow diet since birth, colonized with the microbiota of a conventionally raised mouse, and then separated into Western and chow diets, and then analyzed after 8 weeks on those diets. B and C are 2 other experimental designs that were conducted in this. So B is just showing that they used conventionally raised mice from the very beginning, and given half of them the chow diet, half of them the Western diet, and again on those 2 diets for 8 weeks. And then C is showing the conventionally raised mouse given the Western diet. So conventionally raised, starting off with the Western diet, and then these mice were put on 2 different diets after 8 weeks of being on the Western diet. So one of the diets was the fat restricted, which is the fat R you see here, and then carbohydrate restricted, which is right here. And this experiment was done to show that this can actually be reversible, and it was proven that it was reversible. So going back to experiment B, these results were correlated with the results of experiment A. So Western mice gained significantly more body fat than the chow diet mice, and their microbiome consisted of higher ratios of firmicutes. And back to experiment C, so once these mice were taken off of the Western diet and onto a fat restricted or carbohydrate restricted, they actually decreased their body fat and stabilized their weight. So the end results after sequencing the microbiomes, there actually was a significant decrease in the firmicute ratio, and the bacteroidetes, the ratio of those actually increased. So it did show that taking them off of the Western diet, you could reverse the effects of being on such a high-fat diet. So in another experiment done by Hackett, Corian, Foley, and Erickson, this was recently in 2017, and they tested 16 rats throughout the course of 60 days. So 8 of the rats they used were lean and 8 of them were obese. And these rats were fed on a diet mainly of sugars and corn oil. And they were on this diet for 60 days, as I explained before. And after the 60 days, their microbiomes were tested as well. So all of the rats gained weight on this diet, but the obese rats gained significantly more weight than the lean rats. So as you can see in the diagram next to it, the lean rats started off with lower ratios of firmicutes, and by the end of the 60 days, their firmicute ratio had increased greatly. And then the obese rats had a high ratio of firmicutes to begin with, which is suspected. And their ratios of firmicutes actually remain the same. But like I said, in the results, they gained a lot more weight than the lean rats did within the 60 days. So I keep talking about firmicutes and their ratios within the body. Now I'm going to explain to you what firmicutes are and how they contribute to obesity. So firmicutes are a specific phylum of bacteria that make up the largest portion of the gut microbiome in both humans and mice, which correlates to the results that I have previously explained. So there are 3 different classes of firmicutes. There's Clostridia, which are anaerobic. Then there are bacilli, which are obligate or facultative aerobes. And then we have the mollicutes, which are actually parasitic because they use the nutrients that are within the body, but they are what are most abundant out of all the firmicutes within the gut microbiome. So firmicutes have been proven to play an important role in host energy absorption while in the gut. And their role is a key factor as to why obesity is correlated with high ratios of firmicutes. So what firmicutes do is increase the amount of fatty acid absorption in the body. Also thought to increase the bioavailability of these fatty acids. Now, fatty acids are molecules derived from lipids. And your body, in order to obtain energy, uses the 3 macronutrients from food. So carbohydrates, proteins, and lipids, which is your fat. And lipids actually yield the most ATP per gram in comparison to carbohydrates and protein. So more fatty acid absorption explains why there is a higher increase of fat within the subjects, and also a higher abundance of firmicutes because they are the ones that are using the lipids and breaking down the lipids into fatty acids that are making it more easily obtainable by the body. So essentially the role of firmicutes is to aid in the breakdown of lipids and enabling the host to absorb more energy per gram of fat consumed. And this is why it's hypothesized that higher levels of firmicutes appear to be a hallmark of an obese host microbiome. So in conclusion, the studies that I have explained have shown a correlation between the ratio of firmicutes to obesity. We know after seeing these studies that the microbiome does play a role in metabolism of the host, and having a higher ratio of firmicutes and does correlate to having a higher increase in body fat percentage. So we also have learned that the microbiome can be manipulated, whether this is through diet or possibly transplants of a different microbiome. These different methods could be potentially used to combat obesity within our society and could be a completely different approach to helping those who are obese lose weight and obtain a healthier lifestyle. These are the references I used, and thank you so much for watching.